Thanks, Nicholas. It's my great honour to introduce our keynote speaker for today, Professor Pablo Zarco-Tejada. Professor Zarco-Tejada holds dual appointments in the Faculty of Science and the Faculty of Engineering at the University of Melbourne, where he leads the Hyperspectral laboratory. His pioneering work focuses on detecting biotic and abiotic stresses in crops and forests using hyperspectral and thermal imaging technology deployed via piloted and uncrewed aircraft systems. Professor Zarco-Tejada is at the forefront of innovative technologies and methods for retrieving chlorophyll fluorescence and vegetation biochemistry remotely. His research has been instrumental in linking physiological indicators of plant health to photosynthesis, playing a vital role in precision agriculture, precision forestry, and broader natural resource management. His academic journey spans the globe, from earning an engineering degree in Spain, an MSc in remote sensing in the UK, to completing a PhD in Earth and Space Science in Canada. His previous roles include faculty positions at the University of California, Davis, and serving as director of the Institute for Sustainable Agriculture in Spain. He was also a senior scientist at the Joint Research Centre of the European Commission. Professor Zarco-Tejada's contributions to the field are widely recognised. He's been named as a highly cited researcher since 2019, with over 150 papers published in top international journals. He's an associate editor for Remote Sensing of Environment and the European Journal of Agronomy, and has been honoured with prestigious awards in Spain, the UK, and Canada. Please join me in welcoming Professor Pablo Zarco-Tejada.
Thank you. Well, thank you. Thank you very much, Michael. It's a pleasure to be here today. I'll be talking about an overview of the progress that has been made in hyperspectral remote sensing, imaging, spectroscopy in the past 25-30 years in the context of plant forest health monitoring, stress detection, biochemical constituent retrievals. About 25 years ago, I felt, or I feel now, very lucky because I was involved, obviously in a very small way, but I was involved processing data and being part of the Boreas project from the Canadian side. I was at that time studying my degrees in Canada, and it was for the people who are probably younger and they are not involved, or they were not involved in this type of project funded by the US and in collaboration also with Canada. It was a large-scale international experiment in which there were specific objectives in terms of fluxes, exchange of relative energy, and the improvement of simulation models. I'm talking about years 94, 96. From the remote sensing side, there were some specific objectives identified. We were talking at the time of biophysical parameter retrievals and scaling up from leaf to canopy. And the main reason was that at that time, obviously, we were all working with vegetation indices, but we needed to understand the biophysical processes behind and how remote sensing could be used using physically based models. But more important, how to scale up from the leaf level to the canopy level, because there were different methods, and I will go through some of them. My involvement was from the Canadian side, using one of the smaller planes at the time, but NASA with Avery's and others were involved collecting data. And we collected data with the imaging spectrometer from Canada, the CASI sensor, and we could demonstrate at that time and make progress on how to go from surface reflectance in large mosaics in the northern and southern study areas of that Boreas project into what we started talking about plant functional types. And that approach was done using red edge spectral parameters. In other ways, transforming the hyperspectral data in a way that we didn't really fully understand because there were algorithms that were empirical, but still allowed us to assess plant functional types and to start talking about the fact that we could identify different aspects related to functioning. It was identified a number of things that needed progress. There was a need for advancing models and technology to quantify biophysical parameters, to quantify actual parameters that could be used. Need for physically based models compatible with forest structure because of the heterogeneity and because of the complexity of the canopy. Some of the models at the time were developed and used successfully in crops, in uniform crops, plain parallel uniform canopies, very simple. And need for hyperspectrometry to advance physiology mapping, I mean, understanding the physiology behind the remote sensing data that we could measure and capture. In parallel, I was involved, and I feel very lucky as well, what was so-called the Bioindicators Project, and that was funded by Canada, and it was actually in Sault Ste. Marie, that was mentioned earlier today. And that lasted for a few years, and the main objective at the time was, okay, we have hyperspectral sensors, and we need to identify physiological indicators of forest condition. How can we do that? And talking about early detection of stress that could be used for actions before damage happens. And that was in the abiotic context. A number of reports and papers came out, but the important thing I wanted to highlight is that we already talked about some functional measures, including, for example, chlorophyll fluorescence. Chlorophyll fluorescence was an indicator or a trait that we didn't even understand at the time. Again, I'm talking about years 1997 to year 2000. And we could see the effects of the chlorophyll fluorescence emission on top of the reflectance, or we thought we could see it, but there were no models, there were no demonstrations that was possible to even use it. And how much was noise, how much was actual effects of chlorophyll fluorescence on top of the reflectance or the radiance data. So there were some limitations and requirements that were identified coming from those few projects and others that the international community, I'm not talking only about our work over there, but that I like to go through. There was a need for mapping chlorophyll concentration and other pigments in forest canopies. And that was important because that was the links that were thought at the time with photosynthesis, stress, and nutrition. Other pigments that we were also interested, photosynthetic and non-photosynthetic, like coranoids, xanthophyll, xanthocyanins, are also very important and interesting to quantify, but it was not possible at the time. We didn't have models, and we didn't have the tools to do it properly. It was critical to determine and to model properly heterogeneous forest canopies because of the complexity. There were no 3D models available yet. Some of the hybrid models started to come out, mixing plain parallel uniform simulations with canopy, with density, planting grids, and so on. But there were no 3D models, or they were at a very early stage. The DART was at the beginning. DART model was at the beginning. Chlorophyll fluorescence, solar-induced fluorescence, was identified as a very top important indicator because of the link with chlorophyll with photosynthesis, but the retrieval methods didn't exist. Some of the early papers show that there was a relationship between canopy reflectance and chlorophyll fluorescence, but there were so many questions about it. I remember having diurnal experiments flying the CASI hyperspectral sensor at different times of the day and showing how photosynthesis and chlorophyll fluorescence were tracked and agreed, but we didn't really believe it. I mean, we were still at that time that we didn't know if that was publishable. We didn't know whether that could be really chlorophyll fluorescence. And operational methods were not possible. Sensors and platforms were not available. So I was really happy when we could fly a hyperspectral sensor once a year. While now, as everyone knows, we try to fly and to collect data, particularly using drones, more frequently. There were key areas, and I will go through all of them. I'll go fast, but because I want to give an overview of the key areas that needed progress and how in each of those we made progress in the past 20 years or 30 years, particularly in those that you can see there. In scaling up methods, we had to develop simulations and models that would allow us to assess not only broadleafs, but also needle leaves due to the optical properties and the structure of the needles. Some of the models were adapted. Some of the models that were developed for broadleafs were evaluated and assessed, and they could be used for needles, but we used carriers and we used integrating spheres to assess the optical properties of the broadleafs. And this was absolutely critical coming from the whole scientific community, because that allowed us to validate models from simpler plate models that simulated leaves as different layers into more complex ray tracing simulations like the ones we see here. The leaf level route transfer simulations were absolutely critical, and they made really important progress in the past several years. The prospect model, the most widely used route transfer model at the leaf level, evolved and made important progress from the 90s, quantifying only chlorophyll concentration and a few other biochemical constituents, and adding in different versions the separation of chlorophyll from carotenoids, adding anthocyanins, chlorophyll and carotenoids, retrieving xanthophylls, and more recently, leaf protein and other carbon-based constituents in versions that are recent in the last few years. So all this progress has been fundamental to understand the photon-vegetation interactions at the leaf level that allowed us to identify and to quantify how biochemical constituents absorb light and radiation at different wavelengths, how different biochemical constituents would absorb radiation coincident in overlapping spectral regions, and how we could separate the effects of the different biochemical constituents on the hyperspectral reflectance that you can see there moving with all biochemical constituents at the same time, although they would vary independently. That is something that was fundamental to understand the scaling up processes. It was important to quantify parameters at the image level, obviously, that was the final objective, but we needed to understand how to link leaf level to canopy level, or needle level to canopy level through physically based models, and accounting for the differences in the canopy structure due to the different species and the different characteristics of the forests. In that sense, models that were developed were also absolutely critical, from canopy reflectant simulation models, very simple, to more complex, including flight or dart, among others, that allow us to simulate the complexity of the canopies. And going into linked leaf and canopy simulation layers, in which at the end we could quantify a number of plant traits from hyperspectral data, linking properly the leaf models to the canopy models. How complex both the leaf models and the canopy models. Depend, obviously, the outputs and is a function of the canopy structure. Another important progress that was needed was to go beyond vegetation indices. If we go back to the 70s, 80s, but even nowadays, we are used to quantify vegetation indices, and they have been obviously a very important success. The success started really in the 70s. That is one of the first papers showing the relationship between canopy reflectance indices and, for example, biomass. And that was published in 1973, and after that, several vegetation indices have been proposed related to different aspects: pigments, photosynthetic, non-photosynthetic constituents, even to structure and so on. And they could be also validated through physically based models that would show the sensitivity of those indices to structure, pigments, photosynthetic and non-photosynthetic pigments. But most of the approaches have been or were always empirical, and nothing against, they work, but they are also limited in terms of sensitivity to other parameters or other aspects that I will go through now. So yes, they work to retrieve and to quantify the relationship between those indices, for example, and biochemical constituents, but we all know that they are empirical relationships that are site-specific. So depending on the species, depending on the phenology, depending on the age and different aspects, those empirical relationships usually fail. And we know that when we try to apply empirical relationships from one site to another, or from one stage, growth stage or phenological stage to another. So there was a need to go forward and to leave behind the vegetation indices and then focus on the actual parameters that could be measured, parameters that could be validated, while indices are non-dimensional quantities calculated from different reflectance bands from the spectra. So the retrieval of leaf and canopy traits was done or has been done by the scientific community using these approaches of linking leaf and canopy models, and they've been quite successful. In the past 20 years, we are now confident about the retrieval of parameters as we can see here, that from forest areas, we can quantify biochemical constituents at the same time if we have the right type of hyperspectral data to do it and the right models to do it. But I think that the objective was or is to go beyond actual parameters. Sometimes it's not enough to have specific biochemical constituents or parameters that are of interest. I think the important is to go beyond and to actually quantify functioning and to go to stomatal conductance, mapping of stomatal conductance, transpiration rates, and assimilation. In that sense, canopy temperature has played a very important role through the links between transpiration rates and temperature that was identified in the late 60s and early 70s, and initially in crops. And there's a direct link between canopy temperature and transpiration rates that, for example, in water and irrigation and water stress studies are applied and are successful. Well, now we are starting to apply that method, and particularly using the so-called CWSI, crop water stress index, that comes from crops, but we are adapting those to forest species, where we can that way normalize by the ambient conditions, relative humidity and deficit pressure, deficit, vapor deficit, pressure deficit, in order to quantify and to map properly the stress levels. In terms of assimilation and photosynthesis, solar-induced chlorophyll fluorescence has played a very important role in the past 20 years. And the reason why chlorophyll fluorescence has become so important is because it's around only 2% of the total incoming radiation, but it's a clear link. and demonstrated link with canopy photosynthesis. And the problems is that it requires high spectral resolution and specific techniques. We are starting now to use dark features, which are called Fraunhofer lines. These are dark lines that are in the solar spectrum, and by looking inside with very high resolution, I'm talking about sub-nanometer spectral resolution, we can look inside each of those dark black lines, and any radiation that is found there is due to the re-emission of radiation by chlorophyll fluorescence. So we are using that approach, using sub-nanometer and narrowband hyperspectral sensors, particularly if we focus on the region here where the chlorophyll fluorescence emission happens. And that is something that we can do with new sensors that have been developed, imaging and non-imaging. Like, for example, this is a sub-nanometer hyperspectral imager that we fly and we operate in Australia, in Melbourne University, that allow us to get very detailed radiance with between 0.1 and 0.2 nanometers full width half max. What you see here is not noise. Obviously, we have a little bit of noise, but it's not noise. What we have here is the absorption at those so-called Fraunhofer lines. And we can concentrate on the O2B and O2A, the oxygen absorption features, to apply the Fraunhofer line depth method to quantify chlorophyll fluorescence. The scientific community has been also moving into whether we need these very advanced sub-nanometer hyperspectral sensors by looking at narrow-band hyperspectral imagers like this one here, which is instead of 0.1, 0.2 nanometers full width half max, we are talking about around 5 to 6 nanometers full width half max, but still being able to look into the oxygen feature with sensors that are very compact and still providing very high quality data. And we can look at the oxygen features like this one to quantify chlorophyll fluorescence, showing in different experiments and publications that chlorophyll fluorescence retrieved and quantified using the oxygen feature with narrowband and sub-nanometer hyperspectral sensors mimic the trends of photosynthesis over the season. In that sense, in the past 20 years or more, SIF, solar-induced fluorescence, has been progressed in some ways using models to allow the accurate quantification of the chlorophyll fluorescence. That is a chart coming from a paper that we published a few years ago. It's a summary of all the work in the past 50 years of chlorophyll fluorescence. And what we show is that over the course of the time, from the 70s to current, and from photosystem level, leaf level, one-dimensional canopy models, 3D models, and land surface models, how the modeling has been progressing over the course of the time, identifying a number of models that are currently used for the quantification of chlorophyll fluorescence and the assessment of photosynthesis using hyperspectral data. SCOPE is probably one of the most widely used, but there are other models, including DART or FluorFlight, 3D models that account for the chlorophyll fluorescence quantification and chlorophyll fluorescence emission. Development of new indicators of early stress, pre-visual, that was also identified as one of the key aspects that needed progress. And that links also with how to disentangle biotic and abiotic interactions of stress. With all the progress that was made with biochemical constituent retrievals using leaf and canopy models, with the contribution of canopy temperature as an indicator of transpiration and stomatal conductance, And with the contribution of chlorophyll fluorescence as an indicator of photosynthesis, the question is how can we use it in a way that we could integrate all these inputs in order to detect stress. And by detecting stress, I mean biotic and abiotic stress. In the context of diseases, we have made important progress in a number of pathogens and diseases, particularly in Xylella fastidiosa. Xylella fastidiosa is the #1 and top biosecurity risk in Australia. I believe it is also in New Zealand. It can infest more than 550 species worldwide. It has devastated olive regions in southern Italy, for example, but it's spread in the Americas and then it arrived into Europe. But it's not only a risk to agriculture, it's also to forest areas as well and natural vegetation. So the objectives were whether we could make early detection versus damage. I think damage is well known, that can be identified and detected using remote sensing data because we are talking about structural effects and effects that are normally visible. It's the early detection and the pre-visual assessment what is more critical from the remote sensing science. And disentangling biotic versus abiotic stress. The methods that the scientific community has proposed, I'm not going to get into the details, but it's linking mechanistic models to quantify plant traits using leaf and canopy reflectance simulation models with machine learning empirical approaches to be able to identify the role that the biochemical constituents play on the disease detection. By using hyperspectral data, the community has been able to identify and screen a large number of traits, being those traits biochemical constituents, but also specific vegetation indices that are related to processes happening at the leaf level that are not captured by gravity transfer models. But in all cases, we have demonstrated in the context of different pathogens and diseases that with hyperspectral data, we get generally over accuracies exceeding 90%, while if we go to multispectral data or standard RGB, we go a lot lower in terms of over accuracies, getting sometimes no more than 60 or 65%. That has been applied to Phytophthora, for example, in addition to Xylella fastidiosa, as I said before, or Verticillium. And we have demonstrated that it's possible to identify the most important contributors of the detection of the stress. Finding, for example, in the context of Phytophthora, that, for example, canopy temperature was the #1 indicator contributing to the accurate detection of Phytophthora infection, and chlorophyll fluorescence, structural and biochemical constituents were critical in the contribution of the detection. More recent papers by Watt and collaborators with the prediction of severity of Dothystroma, needle blight in radiata pine, we see there that how important is to use not only vegetation indices as inputs into the models built for the detection, but how important is to identify model inverted traits plus specific vegetation indices that allow us to do an accurate quantification of the stress induced by Dothystroma. More important, by doing and following these approaches, we identify for early stress which are these bars here, we identify which ones are the most important indicators that need to be used for assessing and detecting Dothystroma infections. And what we find is that blue and green spectral-based indices and traits in the blue-green-red region are the most important indicators for the earliest stress. So combining plant traits retrieved by physically based models plus indices that are capturing processes that are not simulated properly by leaf-level physically based models allow us to identify the early detection and to do the early detection successfully, or at least more successful than if we don't use them. And finally, operational methods. What are the operational methods that are needed in the context, for example, of plantations with nitrogen and phosphorus assessments? All the approaches that I said before can now be applied, and we can evaluate, for example, in experiments with treatments, we can identify what is the main contribution for nitrogen versus phosphorus, for example, of the biochemical constituents which are used as inputs into the models for the retrieving of those nutrients. I always like to show the evolution of the hyperspectral sensors when we talk about operational aspects. And that's the CASI sensor in 1997, and that is what happens just around 15 years later. But that's what happened just a few later after the micro-hyperspectral. That is the nano-hyperspectral sensor with 300 spectral bands at 5 or 6 nanometer full width of max, which could be even installed in some of these very low-cost platforms. We flew some of those nano-hyperspectral sensors on these foam-based or foam-made made platforms, which were just a few $100s, while the sensors were 30 or 40 or 50,000, which didn't really make sense, but it was important to demonstrate that we could actually do high resolution hyperspectral data collection using this type of platforms. In order to have a little bit of discussion, and I'd like to go now to the conclusions and final remarks, because not everything obviously is positive, and there are some aspects that obviously need improvements and progress. I think that it's quite clear that the progress made in the past 20, 30 years with hyperspectral data has been very successful, and there are ways to detect biotic and abiotic stress using remote sensing hyperspectral imaging spectroscopy. Development of route transfer models, so we don't rely on empirical methods, which are very site-specific and cannot be applied worldwide or globally. Progress in the stress detection, both in the context of diseases and also nutrients and water stress. I think that it's important progress on leaving behind vegetation indices, going into traits, biophysical parameters, and then functioning. We are talking about being able to map and quantify photosynthesis and transpiration rates and stomatal conductance. And sieve and canopy temperature obviously play a very, very important role when we are trying to model photosynthesis using remote sensing data. And I think that is clear that when we are trying to build models that work and can be adjusted to different canopies and different species, we need physically based models. We cannot rely on empirical approaches that are, as I said before, site-specific. We need to be able to model and simulate processes that can be transferred, and also to, in the context of diseases, for example, to add new databases to existing databases to allow for expanding and making global models for the early detection of pathogens and the stress induced by pathogens. I think that most of the push in the past 30 years has been on the technology side. I think that is the so-called technology push that has allowed to move very fast in terms of the technology available. The miniaturization of hyperspectral images is being obviously clear, and those are the pictures I showed. And obviously the drones, the platforms that can be used to collect the data. But we need to ask ourselves the question, which is how much has been really adopted outside research labs and outside university environments or institutes. What is the adoption of service providers? Sometimes we need to collect data in different parts of the world. I think it happens to everyone. I'm talking about airborne or drone-based, hyperspectral or thermal, and it's extremely difficult. It's very, very difficult to get good quality data provided by service providers if we are not the ones collecting that data. So I think we need a bit of criticism about it because yes, the technology is there, the methods are there, but the transfer to the service providers and the and vendors or the access to the data with quality is not there yet. Drones obviously have been a tremendous progress. And I mean, 15 years ago, we were already flying some of those hyperspectral sensors on board drones, fixed wing, electric. And that was amazing, yes. And I don't think that anyone argues against. But I have to say that with drones, we are going backwards sometimes in terms of what we can really do in terms of remote sensing. And I'll give you a very quick example. Sometimes I hear, no, we cannot fly a hyperspectral, or we cannot fly a thermal and hyperspectral. We can only fly a multispectral or an RGB because the drone doesn't carry or doesn't have the payload that we need, that is needed. Well, what are we doing? We are in 2024 already, and we are talking about that the only thing we can fly is RGB or multispectral cameras. I think this is really going backwards. In the 80s, 90s, in the early 2000s, we collected hyperspectral data much easily than sometimes we do now, because we were not imposing the limitation of what a drone can do. So I've been, and I am, very supportive of drones. We use it in our labs and in Europe, both in Europe and in Australia. But sometimes I feel that they are a real limitation in terms of payload, in terms of what sensors we are carrying, and in terms of a scale. When we need to fly 2,000 hectares, the answer is no. Well, then I'm not interested. Anyway, there's a bit of discussion about that, and I know that the scientific community talks about it also. I think that, well, the alternative, why don't we use hyperspectral satellites? Sure, and there are very good examples available right now, and we can use them. The problem is that for damage mapping, for early detection of stress, sometimes we need to focus on very high resolution targeting pure tree grounds. And that is not possible with the satellites that we have available right now. We are talking about a maximum of 10, 20 meter pixel sizes. Because in some cases, we need to still go to sub-meter spatial resolutions. We normally fly 40, 50 centimeters. I don't think we need 1 centimeter resolutions for most of the things that we do, but at least we need to identify pure tree grounds, because by identifying pure tree grounds, we can quantify processes at the tree level. So the contribution of hyperspectral satellites, I think, is mostly on damage mapping, intermediate to advanced stress levels. For the early detection of stress, we still need very high resolution, sub-meter spatial resolution, both in hyperspectral and thermal. And thermal, it's extremely important when we are trying to deal with stress, biotic and abiotic. And the problem is that still, And again, we need sub-meter spatial resolutions if we want to do a good job. Because sometimes we are talking about 1 degree difference between a tree that is infected and a tree that is not. Or water stress as well, we are talking about a couple of degrees sometimes that it's totally removed, those effects, by structural background, shadows, and other effects. And finally, I think that there is a need for making progress on quick turnaround times of hyperspectral imagery. I think that the dream 15 or 20 years ago that I used to say when I gave a talk at that time was that I wanted to have 1-day turnaround times for multispectral and thermal data. I think we are obviously there. Since we fly, after a flight is done, multispectral and hyperspectral data, multispectral and thermal data can be processed in hours. So same day processing times is easy. Same day processing times for hyperspectral data, considering that in most cases we fly with low-cost hyperspectral technologies. By low cost means that we are not flying a $1 million instrument as we used to fly 30 years ago, makes important issues in terms of the IMU that we used, in terms of the GPS that we use, in terms of the lenses that we use. And that only creates issues in terms of how automatic can be done the processing of the hyperspectral data, particularly if we are talking about processing or flying thousands of hectares, which in most cases is what we want to do. I think I will leave it there. That will be all. I tried to give a summary of our progress in the past several years in terms of hyperspectral, biochemical constituent retrievals, and different aspects. That will be all. Thank you very much, and happy to answer any question.
Thank you, Pablo, for that great presentation. Are there any questions from the floor? Russell, could you please come up to the mic?
Thanks, Pablo, for taking us on that journey. That was really interesting. I wondered if you could comment on the collection of field validation data. Do you see progress in the sensors that are necessary to collect some of these sensitive parameters that you're talking about? You know, gas exchange systems, sat flow meters, or is there the same progress being made, or is the hyperspectral system or sector sort of leaving that behind, or are they in a similar trajectory?
I think that the progress in terms of field validation data has been also great, and we have subflows and subflow instruments, for example, that collect data continuously in trees that we can use for assessment of indicators that we develop for stress detection, transpiration rates, and that is something that we are doing currently, both in agriculture and in natural vegetation as well, and it's going forward quite well. What I feel is that the validation and the auxiliary data that is needed for obtaining good quality hyperspectral data is a bit lagging behind. What I mean is that I think that we still rely too much on targets in the field that need to be deployed every single time that we fly. I don't think that is operational in some way. When I got into drones, I'm talking about around 2005 or so, because I was coming from the airborne remote sensing science community, flying full-size planes and Averies, CASI, and other hyperspectral sensors. What I tried to do was to apply or to transfer the same methods of processing data and data collection. So it was more atmospheric-based data, what I normally collected, which in some ways is more doable than if you have to deploy targets every single time that we fly over an area, because it means that collection is more complex, more difficult, and sometimes it's not even successful. I think that, again, vendors and companies providing hyperspectral instruments provide methods which are simple, but not necessarily the best in terms of science and the quality of the final data that we get. I think that more progress in terms of automatic data collection at the time of the flight is needed, so they could be embedded into the processing that is more automatic without needing every single time to be there while we fly.
Thanks, Pablo. Are there any other questions from the floor or on the app? I've got one on the app here. With the technology push and rapid advancement of drones and cameras, do you have a recommendation for what sensors a new lab should invest in?
What a new lab should be based on?
Should invest in?
Actually invest on? Yeah. Well, it depends on the objectives, I would say. I mean, sometimes when we operate hyperspectral sensors and thermal, I mean, it's a bit of a joke what I'm going to say, but it's true. Sometimes I get people approaching saying, can you fly everything? And I say, well, what for? I mean, what do you need? No, I want everything. I want the complete set. I think that it depends on the objectives, I would say. If we are interested on water stress, for example, if we were talking about agriculture in some forest areas where the main objective is water stress detection, I think that we could just fly a thermal sensor and that's it. And we are going to get very good results. If the objective is nutrients, if the objective is diseases, if the objective is stress, then I would say that we need hyperspectral and thermal sensors. If the objective is more operational remote sensing, I'm talking about someone who needs thousands and thousands and thousands of hectares. I think that trying to do that with a drone is obviously not feasible. And going into multispectral cameras where we can select the spectral bands that we need, it would be a good way to go. We've been quite successful selecting band sets. We don't really need 200 bands in most cases. We can select the right spectral bands at the right spectral regions, depending on the objective that we have. If we are aiming at, for example, pigments, or if we are aiming at stress detection because of a particular disease. So I think hyperspectral for validation of models, for developing, for understanding processes behind that we are trying to develop and evaluate is obviously the way to go. If we want to do it in a more operational way, I think that multispectral cameras where we select the spectral bands would be the best way to go.
Pablo, for that great presentation and comprehensive answer. We have some gifts for you here.
Oh, great. Excellent. Thank you very much.